we're going to continue this conversation with two remarkable people. You saw the introduction, Jennifer Doudna and Eric Topol, giants in the field, doing extraordinary work. Maybe a place to dive in. Jennifer, since the last time you and I spoke, we've seen the example of baby KJ, who was born, an infant was born with a very rare disease. I think CPS1, I think, is what it was. And which is a sort of very rare kind of genetic mutation. In fact, initially when I saw the news, I thought it was a missense mutation, but it wasn't. It was actually one of these nonsense mutations. Maybe you can explain the difference there. But I think if it had been a missense mutation, they could have done base editing. But because it was a nonsense, this other kind of mutation, they had to use CRISPR. Can you talk about that and maybe explain what all that means for us?
Well, it's great to be here back at Zeitgeist, and exciting to talk about this wonderful example of a baby who was diagnosed shortly after birth with a very rare, extremely rare genetic disorder, as you mentioned. It meant that he couldn't digest protein. So he was in the intensive care unit. He had a very poor prognosis. And what happened was truly remarkable. His clinicians at the Children's Hospital of Philadelphia recognized that they had a situation that could be fixable with gene editing. They quickly were able to team up with people across the country, both companies and academic groups, to diagnose the disorder. As you mentioned, it was a single nucleotide, a single letter in the code of one gene that had gone awry, and they actually used a form of CRISPR called base editing to make the corrective change. And that form of CRISPR was invented on the spot using off-the-shelf capabilities. It was delivered first into animal models, both mice and monkeys, to test its capabilities to make this corrective change. And then in conjunction with the Food and Drug Administration, the FDA, making sure that it was being tested appropriately for use in the baby, it was then given to him 3 times, 3 treatments, where the delivery was to the liver, which was the tissue affected by this metabolic disorder. And that meant that within a span of 8 months from initial diagnosis to the end of the therapy, this boy was treated so effectively that he was released from the hospital. And you probably saw the pictures. He is a healthy, happy child. And we never want to say cure because it's, we have to wait and see. But it certainly seems like a very effective treatment for this incredibly rare disease. So a real moment of triumph for CRISPR. That's
incredible. I mean, it's an incredible sign of how this kind of precision genomic editing can make such extraordinary differences. But you're taking that to the next level. I should have actually congratulated you, by the way, that your institute that you founded is now 10 years old, doing all this extraordinary work. So congratulations for that. But I thought you might extend that to, you're starting to do this extraordinary work on the microbiome, which is, I think the microbiome was several trillion cells of everything from eukaryotes to fungi to viruses. But talk about how that's going to kind of further revolutionize the kind of precision medicine and that you can do.
Well, let me just segue from KJ to the microbiome by saying that you might be thinking, well, that's great for one child, but how does that affect the rest of us? How does that lead to any kind of scalable therapies that could come from gene editing? And so I think about this in a couple of ways. One is that I think that the example of baby KJ showed all of us, including me, that this is actually possible, right? And this is something we've dreamed about for over a decade now. It's possible to create a bespoke therapeutic for an individual that treats their disease at the source, and to do it in a meaningful timeline. It didn't take 10 years or 5 years or even a year, right? It was less than a year to do that. I think the challenge now is to do that in a scalable way. And so we're, with our institute, Innovative Genomics Institute, and partnering with the Chan Zuckerberg Institute, we're trying to scale this up in initially a small way that we hope will pave the path for future reductions in cost that could make this a truly sustainable approach to rare disease. So it's a really exciting vision. It's, of course, risky, but that's what we do, right? We take risk and we try to make sure that we're always pushing the envelope with technology. Secondly, you mentioned the microbiome, and I'll just say there that, so the microbiome is, and I'm curious, I'm going to be curious about Eric Topol's view of this, but it's come to, I think it's really come to the attention of scientists and clinicians only fairly recently, that the microbiome, which is all the bacteria that populate our bodies and our environment, plays an incredibly important role in our health. And so imagine being able to use precision genome editing to make modifications to not random bacteria, but individual species that have impacts on our health, and not those entire species, but individual genes within them. And that's really the precision that CRISPR offers. And so we're excited to be doing this both for healthcare applications, but also to address some of the challenges of climate change.
Yeah. Eric, I don't know if you want to add to the comment that Jennifer made on the microbiome, but I had a follow-on question for you. You want to comment on the microbiome?
I think what Jennifer's alluding to is that the gut talks to the brain and the immune system. So whether it's a GLP-1 drug, working on a metabolite of bacteria or bacteria itself, this is a way that we're able to intervene or prevent diseases like we've never seen before. So that's why the work that Jennifer's doing, for example, to try to prevent asthma through the gut microbiome, like who would have thought that was possible? But it's phenomenal work, and I think it has a really high chance for success. I used to say the way to, we know the adage about the way to a man's heart is through his stomach. Well, really what it is the way to human health is through the gut. The fastest thing that's occurring right now, operationally you could say, is that instead of what we used to do in a clinic visit, we now have an automated note that's done from the conversation, which is a whole lot better than the notes that are in the electronic records. That note, which not only is for the patient, so they can go back and understand what was really discussed, and it can be put in their educational level or their cultural language, whatever. But beyond that, the ability for that note to drive things like pre-authorization, to schedule appointments, procedures, lab tests, nudge the patient in subsequent days about what was discussed during the visit, like what were the blood pressure measurements that you were going to get, and how's the physical activity increasing. So that automation of notes is having a big impact, and it was just reported in 6 health systems had a marked improvement, not just for the clinicians, but also for patients. So this is going to be one of the biggest things to rescue this data clerk problem that we have in medicine. But the other things, of course, that are happening concurrently, the ability to take all of a patient's data through many different providers, electronic records, and the corpus of the medical literature, and have that all ready so reducing so much time of reviewing records and not missing things. And then, of course, the accuracy of image interpretation. These are the things that are happening right now so that any scan, things wouldn't be missed. And so these are big jumps that are here and now, and I think this is already shaking up a lot of the health systems, not just here in the US, but throughout the world.
That's extraordinary, but I also want to come back to, I think, some of what you were both talking about before, about this idea of these precision techniques like gene editing and so forth. I mean, currently those treatments cost a lot. They cost millions. Are we ever going to find ways to somehow change how that works? I don't know, through different delivery mechanisms or different ways to do that so that they become more accessible to everybody?
Yes.
Okay. How are we going to
Absolutely.
do that?
No, it's just going to happen. It's going to be a couple of things for CRISPR, at least, maybe multiple intersecting changes. But one is, of course, reducing the cost of manufacturing. And this is one thing that we learned in a very interesting way following the baby KJ case, was working with a manufacturing company. They, in real time, made a clinical grade version of CRISPR that could be used in this child. And in doing so, it's a very expensive process, but they realized there were lots of parts of that process that could be streamlined and streamlined safely. And so I think there we're seeing opportunities to reduce the cost by probably at least 5 to 10-fold. So that's great. And then the other thing, you mentioned this briefly, is the technology and the kind of the science behind it that's going to be required for ease of delivery into patients. This is, in my opinion, one of the big bottlenecks right now in CRISPR technology is the way that we get these molecules into the right cells of the body where they can have the effect that is necessary. And in the case of KJ, fortunately, we had an off-the-shelf lipid nanoparticle technology that was developed in part for the COVID vaccine that could be used to take CRISPR molecules into that boy's liver where they were needed. But imagine that you wanted to do editing in muscle cells or the brain or the kidney, something where today we don't have good delivery technology. That's a very, very active area of research right now, both at companies and in academic labs. So I think breakthroughs are going to be forthcoming in the near future in terms of doing editing much more easily in different cells of the body.
So we'd have a pill or an injection of some sort of delivery, perhaps?
That's the plan.
Yeah. Well, Eric, you've been at the forefront of talking about this idea of expanding health span.
Yeah.
And in fact, you wrote a very influential article in the New York Times early this year about the need to focus on health span. I think a big part of that also had to do with pre-screening, early detection, and those kind. But say more about why health span matters and how to think about it, but also what some of the techniques and things are going to need to be developed.
Yeah, this is the thing I'm absolutely most excited about, and I think is going to be the most important thing that AI does for healthcare. Detection is too late. We want to prevent these diseases. And we have a newfound capability of doing that. And that is by having multimodal AI of all the layers of an individual's data, not just the traditional ones, but also including genomics, proteins, biomarkers. When you have all that data and you know that the 3 big age-related diseases, Alzheimer's, neurodegenerative diseases, the cancers, the common cancers, and cardiovascular, they take 20 years to incubate in our body. Most people don't realize that. Right. And in those 20 years, we should be able to find the high-risk people, and instead of drugs that are used for treatment, this would be on top of lifestyle things, right. We would have preventive drugs. Right. And so this is why I'm really optimistic that we can extend health span, and indeed, we just got funded to start, hopefully very soon, the first prevention of Alzheimer's trial. And that relies on things like p-tau217, the breakthrough test, the biggest things that happen in neuroscience in decades that most people have never heard of. But that is modifiable. And the other big thing that's making this a reality is the so-called organ clocks. So it turns out from 11,000 proteins in the blood that you can get in a tiny blood sample, you can partition and track each organ of the body, brain, the immune system, which is the key to health span expansion. Right. You got to have a healthy immune system. It's very hard to have cancer that can spread in the body if your immune system is intact. And we never had a way in the clinic to measure a person's immune system. But now we have these organ clocks for every organ in the body, the brain, heart, immune system, et cetera. And being able to check that gives us a whole newfound ability to get ahead of the disease before it ever, detection, early screening, that's great, but it's finding the high-risk people before they ever have these diseases. And I think over the years ahead, hopefully we'll be successful with Alzheimer's, we'll be able to make a big dent in these diseases with prevention.
So say more about that, because most screening that most people get is typically in the 40s and 50s and older, but you're talking about much earlier.
Yeah, no, I mean, what people are getting total body MRIs today, which is crazy. Right. Because by the time you see a mass, it's true, a nodule somewhere in the body, that's billions of cells. No, we want to get there at the absolute worst when the cancer's still microscopic and getting the immune system revved up so that it can quash this cancer. So, yeah, I mean, I think that the blueprints are there now, It's the most exciting thing I've seen in medicine, and I think we just have to, follow these leads and get the validation that we can do it. Right. And it can't be expensive, just like you were saying. Yeah. But these tests, these proteins, a genetic sequence, whole sequence is less than $200. Polygenic risk scores are less than $50. These are not expensive things. p-tau 217, also less than $200. So we have new ways. Unfortunately, a lot of people don't know about it yet.
Right.
Yeah.
Yeah, no, it's been extraordinary to see, for example, the kinds of things you can detect with techniques like diabetic retinopathy, for example, which is an extraordinary thing that what you can observe with that. And AI has been massively helpful in being able to do those kinds of detections. But I'm also curious, Jennifer, from a biomedical research point of view, how are biologists and researchers using AI to advance their work? And are there capabilities, are there instances where it's extraordinarily helpful, where AI is not helpful? And also, if you maybe say something about, are there some missing capabilities in AI that you think, if developed, could be massively helpful in doing the kind of work you do and your colleagues do?
Well, I would say in our experience with research, number one, AlphaFold. Thank you, Demis.
Yeah.
It's incredible. We use it every day. Most people I know who are doing biological research use it every day. It's just an incredibly valuable tool. A close second is coding, because a lot of biologists who are, people like me who are experimental biologists, we're not trained as computer scientists, and we haven't had much experience coding. And so I use it, and my students use it for writing, fairly straightforward code that would take them probably many hours if they had to try to do it themselves and troubleshoot it, versus a few minutes if they can prompt a chat type program to give them code that allows them to analyze experimental data. So those are the two ways that we're using it daily. And then less frequently, and I think this aligns a little bit with what we've talked about with analyzing data that are being used to generate reports and manuscripts. It's very useful for that type of thing as well. But I personally don't use it that often. I should. I clearly should, but I don't. Yeah. So
in other words, vibe coding is not just for software engineers, but for biologists too. Maybe.
Absolutely. It's incredibly valuable. Yeah.
Eric, I want to touch on something else you also wrote about recently in the New York Times. About in the practice of medicine itself, and how a lot of doctors and practitioners are starting to use AI in the work that they do day to day. And there are some surprising findings in that work you reported on from different studies. Can you just describe that a little bit and summarize what you found?
Yeah, James, that was really a big surprise. So there were 6 studies that compared the performance, the accuracy of AI versus doctors with AI, where the AI was better than the doctors with AI. That wasn't predicted, no. Yeah. And so we don't really know why. It could be because doctors have an automation bias, and also they may not be grounded, they really know how to use AI. Right. Or it could be that, this is kind of like Waymo, where you're getting superhuman performance that really can't be matched. So we're going to be keeping a really close eye on this with more studies. The problem with most of the AI studies in medicine to date is they aren't real world. They're, with actors that are patient actors, or with, sort of contrived questions or case scenarios. So we have to see how that works when we really use it in the real world of medicine, which is a lot more complex and messy. But it does tell us something that we not had expected. We always thought there would be this hybrid capability that was enhanced with the two together.
Yeah, no, it's actually interesting, because my colleagues in our health AI team were actually building this prototype, I think you're aware of, called Amy. And we're actually doing experiments right now with practitioners at Beth Israel at Harvard, and also at Houston Methodist, to see how much more effective practicing doctors can be actually using these tools. So those studies are still ongoing.
And the other shocker with Amy, and now replicated by many others, is that the empathy is far better through the AI than through the doctors. That wasn't expected. The AI doesn't know what empathy is, but it is able to channel it. And that's a striking advantage.
I want to ask a question to both of you. I mean, you've both been such incredible pioneers in the field for many, many years. I'm just wondering, are there things that to most of us may sound like biomedical science fiction at the moment, but you actually see them possibly happening in 5 to 10 years' time? What are some ideas like that that come to mind?
I guess for me, the one that comes to mind is using gene editing as prophylaxis, protection from future disease rather than curing disease in the moment. And the example that I think is very exciting is actually protecting people from cardiovascular disease, being able to detect by gene sequencing and probably other diagnostics those of us that might be at risk of future cardiovascular events, but because of our genetics, and then being able to tweak 1 or 2 of those genes using CRISPR that give us the protection from that in the future. And that used to sound like complete sci-fi to me, but there are actually now companies that are working on this, and research in laboratories using CRISPR has shown that it's very possible to both detect and change the sequences of genes that do predispose certain people to cardiovascular events. So I think it's coming. I don't know if it'll be 5 or 10 years. I hope it is.
Right. Eric?
I share Jennifer's enthusiasm for a one-shot early in life for people at high risk for heart disease. I think it's just a matter of time that we get there, and that's going to change how we take statins and other drugs. But the thing that has really wowed me recently, out of Northwestern, Shana Kelly's lab, people, I think, have a familiarity with continuous glucose monitors.
Right.
But that's CGM, this is CPM. And you can measure any protein or group of proteins in the blood continuously.
Oh, wow.
So if you want to follow the inflammation in your blood, the immune system in your blood continuously through a patch, just like a CGM, wow, that is going to shake things up. Because when you get continuous data, as we've learned for glucose, how it can help for people to help prevent the progression of diabetes, no less those with diabetes, this is going to be really important because these 3 age-related diseases, the common thread is that it's the immune system dysregulation or loss of protection, which then activates the whole inflammation cascade.
Join me in thanking these 2 incredible colleagues.